So we're just going to give you a little bit of an update on our research following on from Amy now. So, we do a lot of research. Most of the research in our group is on small vessel disease, but that's not just CADASIL. It's also small vessel disease, the very common small vessel disease that affects a lot of the population, causes 25% all strokes. It causes a lot of cases of dementia. But we do have a particular interest in CADASIL, as you know. So in CADASIL, what we're trying to do is look at a number of things. So we look at clinical questions. So, what are the problems in CADASIL? Many of them have been problems you've identified, and say, how can we look into them in more detail to see what causes them, how we can treat them. But we also want to do sort of more detailed analysis of what's actually going on in CADASIL itself. What are the molecular mechanisms, so that we can try and develop treatments which require better understanding of what's going on. So much of our research is built around this register, which has data from about 600 people with CADASIL. This is all sort of collected anonymously, so none of it's identifiable, and we don't use things like date of birth on it. We just use ages on it and so on, so people can't identify people. But it's been collected over a long time, which gives us really a lot of power to answer questions. So I think this is the largest collection worldwide. And once you get to this number, you can then really do statistics on it, which are much more powerful. And particularly if you share it with people, share with other people, then you can get even more powerful data. So we've got clinical information, we've got original MRI scans. We don't share the original scans, but we just analyze them and share the outcome from the analysis. We've got cognitive testing, we've got long-term follow-up, and we've got blood stored. So it's a really powerful resource. I've already mentioned to you about Pat's paper. I think this was a really important paper, but it's only possible because there's so much data there that everybody's given. So it really enables us to answer these questions about, what the prognosis of CADASIL is. This has really provided very definitive data. So, people like other neurologists or genetic counselors and so on, or families, if they want to look at it, can look and see what the average age of onset of stroke is, average age of onset of dementia. So these are sort of little mountains, and you can see, it shows you how many people have got very small numbers of people have got a stroke at about 30, but the most common age of stroke is about, the highest mountain is about 60. What it showed us, which we had suspected, is that the cases of CADASIL we're getting seem to be less severe. So this is a plot of the age at first stroke. So you can see here, this is the age of first stroke, and here's the years against the year of recruitment. And you can see that over time, the age of first stroke is going up. So it looks like people are having stroke at a later age. Now, this could be due to many things. I'll come on to this in a moment, but that's very encouraging. But it does look as if it's at a later age. We can do this in other ways. So we can do what we call a survival plot. So this is where we look at people at different ages, so 0 to 80. We say how many people are free of stroke. So 1 means everybody's free of stroke. We don't get down to 0 because not everyone will have a stroke, but 0 is everybody's had a stroke. And then so you can show when people have their strokes. And what's interesting is there's a clear shift. So the blue line is shifted to the right. So people are having their strokes later. You can see there's about a 10-year difference in onset. So, one of the questions for us has been, why is this? Is this actually because we're seeing milder patients? Is it actually that the disease itself is getting better? And that could be quite a difficult thing to work out. This was published this study was published in quite a high impact journal. It got quite a lot of social media interest and things. So this was a summary that the journal wrote about it. So initial reports suggested stroke occurred in 40s. Perception of many clinicians is the disease may be becoming milder. So what this study adds, patients recruited since 2016 had a lower risk of post-stroke and dementia. And also we looked, I'll talk this a bit later, at what factors might alter this. And for example, smoking had an effect on what effect on this change over time. Because over this time, for example, we've seen a big change in less people smoking with government legislation and things on it. And then it says how may affect provides risk estimates which can be used when counselling patients on prognosis. and emphasizes the importance of treating risk factors in CADASIL. So here's just a little bit of, why you get these results, but it's quite difficult to work out what they really mean often. So, why is CADASIL becoming less severe? So, one hypothesis could be it's better control of risk factors. Perhaps it's, we know actually the age at onset of stroke itself and the incidence of stroke is going down over time, which is quite marked, and heart attack is shot down by about 50%. So it looks like the fact we're picking up hypertension, better treating it, less smoking, better treatment of cholesterol is improving these diseases. So it could just be that it's that we're just improving these risk factors. It may be that, we're picking up milder patients because, when a disease is first diagnosed, you pick up people who've got more severe disease because they're the ones who go for testing, which is quite difficult to get and expensive. And as testing becomes more widely available, you pick up more people. But much remains unexplained. But I think it is very good news, nevertheless. This shows you, so we were able, I think I've showed you this slide before, but we were able to use this data from Pat's study to say, what affects the severity of CADASIL. And as I showed you before, his data showed that if you smoked, it increased it. If you had hypertension, it increased it. If you had male sex, it increased it. Also, if you had a particular site of the mutation, I showed you this earlier on the website where you can find this out, it increased it. So all those factors seem to be important. But what we're looking at particularly now is whether there are other factors. And one thing we think is likely to be important is whether you've got what's called modifier genes. So that we all have lots and lots of genes, and could there be other genes that influence the effect of the CADASIL abnormality that make it more severe in some people and milder in other people? And if we could identify these, it might offer new treatment approaches. So this is really the study that Amy was talking about. So what we do in these studies is we just take blood, and it's very, very easy to extract blood from DNA. You can get it from a saliva or blood. We find it actually easier to store usually from blood. And you take a very small amount of blood, you extract the DNA, and then you do this technology where you're using a gene array, and you're able to identify genetic markers throughout the whole of your genetic material. So up to a million or markers. And then you compare how common these markers are in normal people versus people with the disease you're looking at. And then you can find the particular genes that are associated with disease. So the way we do this is, we have to get lots of patients to this sort of analysis. So we're collecting these cases that Amy's told you about. We do this genetic analysis, and then we say, which genes are associated with earlier onset of CADASIL, and which genes are associated with more severe changes on the brain, which we use, we segment out. Actually, now this is largely using AI segmentation tools. We can segment out these abnormalities in the brain. So this is the last meeting, which Amy showed you a picture of. As I said, you just couldn't do this research on your own. You have to work with other people, which we're doing now. And we've now got stroke data for over 3,000 cases. And we've got MRI scan data this is also with genetic information for over 2,000 cases. So really, really powerful data. We're lucky this is funded partly from a program grant I got from British Heart Foundation that have funded this project. So, but hopefully by next year, we should have some data from this, I think. So that's one thing. So just to move to a very common symptom, one of the symptoms you mentioned was a major problem in CADASIL was fatigue. And indeed it is. So Amy's been doing some work on this. This was published over the last year. So, many of you may have taken part in this questionnaire on fatigue. And we looked at healthy people without CADASIL. About half of the people with CADASIL get fatigue. So this is quite a lot higher than in the general population. So then we can look and say, what causes fatigue? So we found interestingly, it didn't associate with a history of stroke. I thought it'd be worse if you'd had a stroke. It didn't associate with how severe the disease was on MRI, which was a surprise, actually. But Amy's actually done a lot of work on this in other diseases generally, in stroke in general. We haven't found a relationship between the presence of fatigue and severity of disease on MRI. It was more common in the presence of depressive symptoms, and it was also more common in people who've got some cognitive impairment. But we do see it in people who've got neither of those two things. So it's something that we're sort of exploring at the moment. It's a really difficult symptom. It's quite disabling and difficult to treat. But the good news is there is a trial that we're now part of, but this isn't for CADASIL, this is for stroke in general. This is an Australian UK trial called MIDAS-2, which is looking at a drug called modafinil. This is the drug that causes sort of wakefulness. It's used in a disease called narcolepsy, where people fall asleep just at that, during the day. And a small study suggested that it was helpful in stroke. So this is a much bigger study in about 500 people, I think, isn't it, something like that? It's a randomized double-blind trial. So you take the drug, but you don't know whether you're getting the drug or dummy drug. And then at the end of this period, we see whether people who've been on the real drug have got less fatigue than people who haven't been on the real drug. So if that turns out to be positive, then that will be really useful, I think, for CADASIL as well. And there is talk about doing a specific trial in CADASIL, but my feel is probably the funders wouldn't fund it until they've seen the result of this study, probably. So that's fatigue. So then the question, I get asked a lot is, are there any treatments, really reversing the CADASIL itself? And, there's a lot of interesting areas, but there aren't any treatments at the moment. Just perhaps to go back a moment and say, what happens in CADASIL itself, just so you understand, what sort of things you could target. So I mentioned to you before about the DNA on the left, and the DNA is used to encode proteins. So proteins are sort of the building blocks in cells. And the Notch3 protein sits on the membrane of cells. So if you look here, this is a Notch3 protein here. You can see here, this is the cell membrane. And all of the mutations occur in, all of the variants occur in this part of the protein here, the bit of here. So what happens if you have a CADASIL mutation is you get abnormal Notch protein, which then, because it's abnormal, it sort of aggregates together and forms little aggregates, which you can see stained here in the sort of the dark brown. And this collects in the cell wall and disrupts the function of these small blood vessels. So there are a couple of ways that people are exploring trying to do this. So you could just sort of change the gene function so you make less protein. That's one possibility. The second thing is you could perhaps try and clear these aggregates out. So the first method is to try and change the protein level, is you can actually interfere in the process by which DNA is converted to proteins via what we call messenger RNA. I'll just let Amy So this means you get less Notch protein produced. And actually, these are being used quite a lot in neurological disease, these antisense oligonucleotides. So they're being used to sort of down-regulate production of proteins in a whole lot of different diseases. And the one perhaps which has had most impact is this thing called spinal muscular atrophy. So this is a childhood disease where you get weakness usually during childhood. And they've shown some really quite good results. So this is one approach we could apply in CADASIL. And I think I may have mentioned to you a while ago, there is a company that's quite interested in this, but there are quite a number of challenges with this. One of the challenges is, how do you get these antisense oligonucleotides right deep into the brain? You couldn't give them in the bloodstream. You could perhaps give them in the CSF. You could perhaps, do a lumbar puncture and inject them in the lumbar puncture, but would they actually reach deep enough into the brain? But we're hoping that the company is going to take this up and take this forward. Obviously, these are very, very expensive programs, and the difficulties the company then will assess, can I make enough money out of this to make it worthwhile doing the investment. But that's one approach you can do to alter gene function. The second approach you can do is a thing called vaccination and immunization. And this, so what you do here is you develop a vaccine against the abnormal Notch protein. So this is a protein that's deposited in the cellular wall. And for some strange reason, which I actually find quite difficult to understand, but this leads to clearance of this abnormal brain protein. And some of you who keep an eye on Alzheimer's disease may have heard about immunization for Alzheimer's disease. There was a lot of interest in this, where you could vaccinate against or immunize against the amyloid protein, and it cleared the amyloid protein. It didn't actually work well in Alzheimer's because it didn't seem to be well suited to protein right in the middle of the brain. But in protein in the blood vessel wall, it may be better. So this is the Alzheimer's I told you about. And there are some interesting results from animal models in this. It's not been yet used in humans. But you can show that if you vaccinate against the Notch3 protein, you reduce this abnormal Notch3 protein. So that's another approach, which I think is very promising. But taking these approaches from animal models into humans is really difficult. And there are two things. Does it work in humans? The second is it going to cause side effects? And they've both got to be favorable to take it forward. Alternative approaches. So we could also do other much simpler things. So, can we find drugs we've already got around at the moment that could improve the disease? Can we develop new small molecules? There is actually a trial in Japan of something called adrenomedalin, which is being assessed in 60 CADASIL patients. So there are trials going on at the moment. This is, you probably won't be able to read this, but if it says it's a vasoactive peptide, so it's a peptide which acts on small blood vessels, which has angiogenic, that means it helps blood vessels grow. Vasodilatory, that means it opens up blood vessels, and anti-inflammatory, that means it reduces inflammation. So it's got multiple actions essentially. So it's good that people are doing trials. I'm not sure whether this is going to lead to I'm not sure the scientific basis is so strong for this, but we can always hope. It would be great if it does. And these are some of our collaborators who are working with us from Japan. Here's a picture of Alex, just so you can remember who she is, hiding in the corner. And so what we're interested in is ways we can evaluate some of these new drugs simply, because doing trials in people takes a long time. It's you just don't have time to test lots of different drugs, because in a disease which is very slow, you'd have to do trials for tens of years. So we're trying to develop models where we can test drugs much more quickly. And you've heard Alex talk about this before. This is called the iPSC technology. So this is what Alex does in her lab. So what we do is, and some of you have helped us with these. Now Lupe does these tiny biopsies, sort of a couple of millimeters in clinic. We then take the skin cells. Alex regrows those into cells which are called pluripotent, so they can develop into any different type of cell. She can then make them grow into blood vessel wall cells. So she can make them grow into what we call endothelial cells, these are the lining cells in small blood vessels, and mural cells, either the sort of supporting cells in small blood vessels. So she can get these, put them into a little dish here. Here's the lining cells, here's the supporting cells, and then she can look and see the interaction between the two. And what is really interesting is if you take cells from people who have both CADASIL2 and COL4, you can show abnormalities in these cells in the dish, very clear abnormalities. For example, you can show that they're more leaky, they don't behave normally. And then you can then give drugs to see how you can switch these changes off. So we're very excited that this may allow us to have a way that we can look at different treatments quite quickly, because of course you can do this, very quickly. This is how you can then scale it up. So you can actually look at lots, you can get up to plates that have a number of little chambers in them. You can get up to 96 well plates. So you can do many more experiments at the same time. So this is really what we're hoping to do at the moment. This is Alex's talk on it. So if you want to hear more about it, do listen to Alex talk about it. It's really interesting. So this is also on the website under CADASIL Research. I think it's a talk you gave last year, isn't it, Alex? So it is really exciting technique, I think. So do have a look at that if you're interested in hearing more about it. A lot of this depends on getting the right samples from the right people, because we want to get particular samples we can grow into cells. So we work with something called the NIHR Bioresource. So this is a large resource funded by the research arm of the NHS, which collects people who want to develop volunteer research and then can call them back by specific features. So it can call people with a particular genotype back. So some of you have been recruited to that, and then we can call people back, do a biopsy if we need to, and then derive these cells. But as you know, Lupe now is doing biopsies in clinic as well, because it just makes it so much easier if we can do everything on your one visit. And good news is that yesterday, Alex and I were awarded a grant to take this work forward after more than one attempt. So it's really exciting. So we can now work on this more. We're looking to get more funding on it. And just lastly, Natalia showed you that if we're wanting to evaluate new therapies, we need ways of grading the disease for clinical trials. So Natalia is doing some work on this, but over the last year as well, we've done a big collaborative study, which is led by Sassi Leslie-Oberstein, who's one of our close colleagues from Leiden in the Netherlands, which has sort of involved lots of people, as you can see, with a lot of people with CADASIL, a couple of thousand, and shown that you can sort of grade the severity of the disease using a combination of MRI, clinical features, and other features. So we hope that this will be the sort of thing you could put into a clinical trial to see whether therapies work well. So, just lastly, the research would not be possible in any way if people didn't volunteer to take part in it. So, we really are very grateful to anybody who does take part in it. If you want to be involved, I think I said this earlier, but we have registered CADASIL patients. Amy can tell you anything else you want to know about this. You do need to be registered as a patient on the NHS, but we can take people from the NHS anywhere in the UK. And if there are areas in CADASIL that you feel we should be focusing on, please let us know, or otherwise let CADASIL Support UK know. Then we can, that's where fatigue came from and things. So if there are symptoms you think are particularly important, then we should look at those. I think I've already said this. And I think that's just Amy there. So we'll stop there. Thank you very much.